Welcome to another episode of NatureSpeaks, brought to you by Himalayan Nature. As always, I am Aditi, your moderator for the session, and for technical support, we have with us Dr. Tulsi Sudedi sir, Director of Himalayan Nature. So each week we bring to you interesting talks on wildlife, environment, and many other aspects of nature conservation and its importance in the world today. We have had talks on sustainable tourism, importance of litter decomposition, and conservation of endangered species like vultures, clouded leopards, elephants, and tigers. And today we are gathered to talk about another endangered species, and that is red panda. Known for its cute features, the red panda are a charismatic mammal species native to the eastern Himalayas and southwestern China. They are endemic to the temperate forests of the Himalayas and range from the foothills of western Nepal to China. However, despite various conservation efforts, the population of these species continue to decline due to various anthropogenic factors like habitat fragmentation and poaching. This has led in red panda to be listed as endangered in the IUCN Red List. To discuss more on the conservation of the species, we have with us lead researcher Dr. Arjun Thapa, who was also one of the prime researchers to make the finding of two different species of red pandas. Dr. Thapa did his PhD in ecology in the Institute of Zoology, Chinese Academy of Science, Beijing, on conservation ecology and genetics of red pandas across the entire range. He has had several research experiences in assessing climate change's impacts on species and habitat, assessment of indicator species of climate change in Gandaki River Basin, identifying important river stretches for conservation based on fish distribution in Nepal, and also assessment of red panda along with various other endangered species. He has had numerous publications on endangered species like pangolins, musk deer, and of course the red pandas. Currently, he is the research director of Small Mammals Conservation and Research Foundation, and it is our honor to have Dr. Thapa with us this evening to learn more on the way forward to red panda conservation. Namaste Arjundai, and welcome to the NatureSpeaks series.
Good evening, everyone, and namaste. Shall I start my presentation?
Just a moment, before I let you start with the presentation, I'd like to inform our audience that this session is being recorded. If you have any questions regarding the presentation, please use the chat option, which will be available shortly before the presentation ends. And our speaker will try to answer as many of your time allows. And with that, I'd like to ask Dr. Thapa to start with this presentation, please.
Shall I start?
Yes, please, Dai.
Yes. Good evening and namaste to everybody and everyone from home country or from abroad. Myself, I'm Arjun Thapa, working on Small Mammal Conservation and Research Foundation. Today, I'm going to a little bit talk about a way forward for red panda conservation across the nation or in that range. Today, I focus my presentation on highlights on species, basic information, research and conservation, and future perspective. Going through these pictures, I try to include both important of biological significance and conservation significance on the species. Why red panda is unique, perspective of evolutionary uniqueness, because it belongs to other carnivores, but entirely depend on vegetarian diet, basically on bamboo species. This perspective gives some biological importance why we interest for red panda. From management perspective, the population is declined due to many threats, and it is distributed across the 5 Asian range countries: China, Nepal, India, Bhutan, and Myanmar. With declining population, they are including endangered species in IUCN, as well as they are including epidemic fossil scientists. From these figures, I try to include both biological and conservation significance of the species. based on species highlights. What the previous study gone on red panda, I did all the reviews for 2017. After that, I didn't did. And based on these reviews, we identified habitat, foraging, movement, and molecular ecology is highly concentrated studies. Besides that, pathogen, isotopes, got microbiota. They have a least or limited study. So the area, because area for the future research, maybe focus on these areas will be give new findings. Also based on reviews, most of the previous study are focused on captive, a little bit in wild. Recently they are focused in wild, and most of the research belongs to other category, means this study include in captive, and most of the study focused from China, then Nepal, India, Bhutan, and very limited study from Myanmar. And including a different aspect, most of the study focus on anatomy and physiology, which means that belongs to other country, others categories, and disease, genetics, distribution, ecology, diet, behavior, and many things. These are the inside the category of published information. All the scenario gives study on red panda shift from captive to wild. Before I started my PhD, there is some gaps on the study, and I focus my study for looking this information for insufficient document distribution on potential suitable habitat across the entire range. The Asian red list still didn't include the figures of Myanmar, and even most of the studies are scattered and not well documented. And even model also used previously, models are used, they are, some models are highly precise, some models are overestimated. So I also look for that, and at that time looking, I find that at the gap. And same thing happened with other distribution range. Most of these distribution range after suitability were based on expert opinion rather than fieldwork and ecological variables. These are the previously gap, but I try to address these gaps during my studies. To start my work on red panda, I started with what are the food habits and dietary, do they are dietary specialists, and how much potential habitat suitable for red panda across the entire range. Do current protective area are effective for protecting red panda based on current protected area networks? And what are the important habitat factors associated with red panda? This, Beside this, I focus my study on genetics and population structures. And one of the most important questions need to be solved. Either these species are shrubby species or belong to two distinct species. Not only this, do any paleoclimate influence on genetics of red panda? To answer all these questions, it will focus all the species ecology through the conservation lens, and this evidence will contribute in science-based evidence on conservation. So if we follow these findings for the management, it will be great. So I raised all these questions and addressed these issues or questions in my research careers. For addressing all these questions, many methods are implemented in the field as well as laboratory experiment. In the field, they have now camera trapping, GPS satellite coloring, and transit occupancy survey, quarter samples for habitat composition. In the lab, they will do DNA preservation and isolation, PCR sequencings, pathogenics, macro histological analysis, as well as nutrient analysis. This is the methodology used in context of Nepal currently. In my work, I focus my research in three areas. One, to predict the distribution. Next is identify the important variables and know the level of genetic status of our species. For this work, for predictive modeling, I use Maxent and other ecological and topographical variables to understand how much suitable habitat available for red panda across the entire range. Similarly, for the habitat, I use ground-based surveys, information collected from frangic and quadrates, and do some multivariate analysis and identify the key habitat features. For genetics, I collect the feces sample and do microsatellites, microcontrolled DNA, mtDNA, and microsatellites to understand the genetic diversity and population structures. These are the areas I focus during my work. What are the key findings based on this research? For the food habitat analysis, I collect the sample from Langtang National Park and do microhistological analysis and identified in the all season more than 90% food contributed by bamboo, and which is not significant throughout the seasons, but there are some supplementary food which are very limited amount and they are seasonally vary. Mostly they use some berries like plants. I think this supplementary food might contribute some energy requirement during their breeding or other cases. Through this, we also calculated niche breadth, which is very low. Then we identified consuming maximum foods as well as very narrow niche breadth. Then we said that it is a dietary specialist. For the second findings, I use species occurrence data across the entire range from China to Nepal and build models based on ecological variables, both topographic and climates. And we estimated that more than 61% of habitat lies within China, and then Nepal, Bhutan, Myanmar and India. This current protected nature network only covers around 28% habitat under the protection. It means that more than 70% habitat lies outside the protected area, which indicated there is need of alternative conservation practices or conservation management to protect the more red panda populations. Also, I identified how much highly suitable habitat. Only the predicted 40% habitat falls under the highly or moderately suitable habitat. I mean that 60% habitat on the least suitable. Based on this finding, we also conclude that Bhutan protected area has very good networks for protecting the red panda populations that cover more than 43%. One of the interesting things is that when we did the modeling, we found that both species have different climate space. It means that the red panda that are distributed in the Himalaya are driven by precipitation relevant variables, whereas red panda belongs to China, although it's tiny, that is driven by temperature associated variable. It means that the climate have played key role to make their climate niches across the entire range. One of the most interesting questions is that earlier there was believed to be two subspecies. One is Chinese subspecies, always fulgens steini, that belongs to China only. The Himalayan subspecies, that is always fulgens, that belongs to or that distributed to China, Myanmar, India, Bhutan, and Nepal. With these informations, still there is controversial for that. And they think that the rivers that named by Lujiang River that lies in Yunnan that separate this species, subspecies, to China, to Chinese subspecies and Himalayan subspecies. Already, this information are still controversial due to insufficient informations, but they believe that. So the findings should also go through correcting these biological, these geographical barriers. To address these uh, barriers, or to identify either the Lujiang River acts as barrier or not. In 2011, the Wu et al. Did the study and identified that the 103 genetic population in the Chinese red panda. That research could not address the subspecies issues because they only collect the sample from Tibet, very few sample from Tibet and others from Yunnan and Sichuan, and that did not give sufficient information for subspecies classification. All these research that have some controversial about subspecies classification, and we raise and follow to address this issue during my study. However, they are already known that Gao 1987, Glaston's 1984, and Groom 2011 already mentioned that geographical variation in morphological differences. They have already given these evidences. However, insufficient genetic evidence for the subspecies difference still remains. Then we start our work to collect the sample from Himalaya to solve this solution, to bring the solution. For this task, we use whole genome sequencing, parentally inherent Y chromosome snaps, and maternally inherent mitochondrial genome. We use 3 markers for 65 red panda whole genome resequencing. That includes 44 from Chinese red panda species and 21 from Himalayan red panda species. Now this figure shows that we collect the sample from entire range except this Bhutan and India. However, there is no much barrier for that, geographical barrier in that area. And we assume that the sample from Western Nepal and the east of its distribution will be sufficient for our study. Then we identify these 5 populations for that study. Already mentioned that, already given by Groom 2011, they have already morphological differences on the species. Just looking for their strong frontal convexity, both in Himalayan and Chinese, they have separate in the size, colors, zygomorphic weight, maxillary teeth and rows. They have variation for that. Even they have a distinct tail ring for the Chinese species and blonder or blur ring for Himalayan species. They have the tail ring also have separate morphological differences. Based on these morphological differences, we goes through the source about the genetic differences through the whole genome sequencing. From the collecting information of genomics, we build a phylogenetic tree based on 41 mitochondrial genome haplotype, show the very distinct lineage between Ulrus Folden and Ulrus Steini. It means that the Himalayan species, they have a separate cluster. They didn't share any information with Chinese species. This is the one evidence for species species distribution. Similarly, we did cluster analysis using structure and identified that the Himalayan species did not mix or did not share any evidences with Chinese species from the structure analysis, and we identified 4 population for this species. From these evidences also support for genetic divergence between 2 subspecies. Then we use P-shape plot to know whether they are sharing or not. This P-shape plot also shows that different cluster with Chinese species and Himalayan species. These three information already supported for two distinct subspecies. Similarly, we make haplotype networks for Himalayan red panda clustered together and separate from the haplotype with Chinese red panda, highlight that notable genetic divergence between the two species. It means that these haplotype networks also didn't share anything, or they make separate from Chinese red panda, and even Y SNPs and mitochondrial genomes. On the basis of whole genome SNPs, phylogenetic tree, principal cross-membrane analysis, and admixture results revealed sustainable genetic diversion between the two species, providing first genetic evidences for species differentiations. After the genetic differentiation, we know that what are the major biological possible biological barrier for that differentiations. We searched for that previously. The previous research mentioned that losing river act as a biological barrier, geographical barriers for the species. From the recent study, we identified that Alajambu rivers separate two distinct species rather than the Lujiang River. This Alajambu River is the originate from Alajambu River, actually origination of Brahmaputra rivers. So to understanding the species geographical barrier for the red panda, we believe Alajambu rivers. Based on research, either this barrier could work for other species or not. It should be a great research for other species. Then we can conclude that Himalayan species may be barrier by these Alajambu rivers. Based on these barriers, Chinese red panda include the eastern Himalaya population, whole Galilean population, and the Yunnan and Sichuan population that include China, Myanmar, and certain part of India. Whereas Himalayan red panda include the population from Nepal, India, Bhutan, and the population from South Tibet of China. So we conclude for that Alajambu River is a geographical barrier for this species separation. Also, we look for demographic history for two subspecies. They are traced back approximately 3 million years ago, and the two red panda experienced obviously different demographic history. Chinese red panda has two population bottlenecks and one large expansion that make the Chinese red panda population viable in the wild for the long term. Whereas Himalayan red panda species have three bottlenecks and one very small expansion, it means that the population in continued decline, which the conservation is important for this species to from the genetic levels. From the whole finding, we conclude that red panda are two distinct species, not one. One is Himalayan red panda species, we name it Aulus fulgens, and Chinese red panda we name it Aulus steini. And we believe Alajambu rivers, the origin of Brahmaputra river, act as a geographical barrier of these two distinct species. And all this information have been published in Science Advance in last 2020. From the whole entire genomic information, I want to go through what is the genetic labels of Himalayan red panda in case of Nepal. For this work, the sample is collected from across the entire range from Nepal, from where distribution is known. And we identify from 150 samples, I just identify 40 individuals from the whole samples. And that represents a 13 spatial location from the currently known distribution range of Nepal. From my study, I use For this, I use mitochondrial DNA and microsatellite, 11 microsatellites, and identify low genetic diversity and high inbreeding in this species. Within this, the red panda population in Western Nepal has very low genetic diversity. So conservation should be focused in that areas. Similarly, we will 13 haplotypes networks combined with Chinese red panda. Still, it shows the different than Chinese. So it didn't share any haplotype with Chinese red panda species. Even our haplotype didn't show the good structure across the Nepal. It makes. To understand the genetic structure of the red panda, I use 11 microsatellite and analysis from structure analysis and detect only two population, the western and eastern population is the genetic structures. When I saw the two structures, then I sought what are the drivers to make these genetic structures, and I build paleoclimatic models based on climates, and I found that interglacial climate influenced the red panda genetic structure. From this analysis, we can say that paleoclimate shaped genetic structure of Himalayan red panda. Looking for this structure from my study, I suspect Kaligunrugi is a barrier for this two population structure of Himalayan red panda. What are the importance of findings from my results? What it help to conservations? So from these findings, my findings contribute in developing red panda conservation priority outside the protected area, looking highly suitable area within the protected area across the entire range. Also, we can give some transboundary habitat conservation approach from this research. Also, we can conduct the research in new areas, either they are distributed or not, to find in the landscape to find the new possible sites for red panda distribution based on these informations. Based on genetic informations, what we can do work on connecting for the illegal trades. From this database, from this genetic database, we have the source identity. We have the genetic identity of source population from eastern, central, and western Nepal. If we have the seizure samples from across the Nepal, then we can do genetic analysis and do some structure and phylogenetic tree. Then the sample that is seized from poacher will be clustered with eastern, western, or centrals. So based on this information, we can use for tracking to know the illegal threat and tracking the illegal route mappings. Based on genetic information, we can use this information. I think this will play important role to control illegal trade. And if the poaching sample comes from the entire eastern Nepal, then we can focus our conservation priority for that areas. Based on this, we can do some combination of poach sample and source samples to know the illegal trade samples and mapping illegal route. What is the future perspective for red panda? From my study, I thought that we should look multidisciplinary approach from space using remote sensing, predicting database, and molecules from genetic nutrients, microbiota isotopes, and from ground survey looking from transit, camera trap, occupancy. If we combine all these informations, it we can easily understand the detailed species ecology and phylogeography. This will also contribute to evaluate habitat evaluation, research monitoring as well as population monitoring. Also, there is essential to build a regional database and upgrade the knowledge of ecology on wild pandas. Obviously, it is important to build coordination partnership among the organizations with national and international for red panda conservation. One of the most important issues is to build initiations on transboundary conservation. To enhance the local livelihood, we need to harmonize community-based conservations and monitoring with the local peoples. How can we sustain red panda funding for conserve wild panda population? Our name is Himalayan red panda diplomacy. We know that many Himalayan species, red panda species, that founder belongs to Nepal, India, Bhutan, goes to across the zoo of the world. So they from that zoo, visitor every day pay money to see the red panda through special options as well as direct options. From that option, we can generate money. Is red panda from zoos, then we can build a one basket fund, regional basket fund across Nepal, India, and Myanmar. When we collect the fund from zoo, and it will go to regional fund. From that regional fund, we can separate their own government authorities. From the government authority, that fund will be goes to relevant departments, and that will gives to research and conservation institutions. From that institution, all the monies goes to ex situ and in situ conservation. From ex situ conservation, we can do the red panda breeding or disease or any research on that. In the wild, we can invest for money for research, conservation, and livelihoods. Combining this, if we successfully breed captive breeding, we can give again to global zoos. It's a leads like that is a giant panda. If we work in that way, we can collect a lot of money from zoos and we can conserve our wild panda. This is a simple thinking for that. Thank you for your attention. I finished my presentation.
Thank you, Arjun Thapa, for that wonderful and informative presentation. It was interesting to know about your study and what future we can learn in terms of conserving the endangered species. As you said, it is very important to conserve the red pandas, especially through transboundary conservation in their habitat that ranges across various countries. And we hope this can be done in an international level, along with awareness in conserving the species in local level too. I'd like to go to the Q&A session then. We have our first question from Himsagar sir. And he writes, the taxonomic division has been going on for some time since the paper by Groves in 2011, where he proposed 2 distinct species of red panda. And now yours and your colleagues' studies have given further support to his proposal. So is there any consideration by IUCN to accept these splits and list them accordingly soon? Or if not, what is the story from the other side?
Thank you, Brass, for questions. Actually, we publish the information. Then after that, there is no any IUCN assessment for that. Maybe they will do recently, because the professor who works for red panda is the member of that IUCN assessment for red panda. They think they will work very soon. They didn't do any assessment on IUCN, but already Groom mentioned that they have distinct 2 species. With this, based on that, morphological information, we build some genetic evidences to support that. And recently there is another publication from India. They also support from our research that Alajambu rivers in India, they say Brahmaputra rivers, they also support that. That result also support our results. Maybe this finding will be accepted by IUCN for 2 distinct species very soon.
And to back it up, we have Angela Gladstone, a senior scientist on Red Panda, who has noted that Will Duckworth and herself are discussing rewriting red data listing for Red Panda, I guess. So that is one information to be shared. We have another question from Karan Sahasar, and he asks, do you agree that information on the food spectrum of Red Panda other than bamboo is not adequate? The supplementary food items in Nepal that you have mentioned are deliberate or incidental?
In case of Red Panda, it is not entirely vegetarian. It will be, we cannot say entirely vegetarian, because they have some, during my study, I also found some sample with some skins, not skins, ears, and I think that they eat some other things also. But we said that they mostly they depend upon a bamboo. Still, we don't know which species they prefer, because they have a lot of diversity for bamboo across the Nepal. In eastern, they have different species. In western, they have different species. Besides that, they have some berries, Sorbus cuspidata. They also use seasonally during the breeding seasons. Not sure, but most of the food depend upon bamboo.
Okay. Thank you, Dai. Another question from Kamal Thapa, sir. Do you think there are any genetic exchange in between different subpopulations across Nepal? For example, Eastern and Western, even Langtang and Manang, or within the Langtang subpopulation only?
Based my genetic informations, I identify only 2 structures. I mean that these 2 structures didn't share any informations. They are totally isolated from Kaligandi to Eastern Nepal. They share their haplotypes, and they have that could be separated by recent or Holocene human activities, maybe separation for that. But Western population is already isolated. They didn't share anything with Eastern population. We identified that from microsatellite. They have the clear structures.
Okay, thank you, Dai. Another question from Sam Fassi. With the low genetic diversity of the 2 subpopulations in Nepal, are there plans to mix the genes between the 2 subpopulations? And has there been any investigation into the viability of zoo populations as potential options?
In the wild, I think they did not do any, we do not do any things for maintaining genetic diversity. In case of captive, in our case, we have very few individual, maybe 1 or 2, maybe 2 in our captive. I think they did not do anything. But in European zoo, they do, and they maintain genetic diversity in good levels in their captive population.
Question from Dumbar Bista, sir. It seems like the population in Western Nepal is relatively more vulnerable due to low genetic diversity and inbreeding. So how long do you think this population can survive with existing conservation measures? And what could be the minimum viable population to sustain these in long run?
It is difficult to answer these questions, but the population of, we notice that population of Western Nepal already goes down. So we should focus our conservation intervention in Western Nepal. That is the only signals for that.
Okay. We have a question for the morphological differences between two subspecies. Mr. Nobin Bhattaji wants to know if there are any differences in their habit as well between the two subspecies.
There is a lot of informations from the captive for behaviors. From the wild, we don't have the sufficient informations. One thing is very difficult to find to observe panda in wild habitats, and then it's difficult to do some behavioral study. But if we go through for detailed study, maybe they have uh, some differentiation and barrier, some differentiation in behavior. I don't think so much, but resource also drive their behavior changes.
Okay. We have a question from Suraj Baral, sir. What are your thoughts behind the reason for low genetic diversity, especially in the western part? Do you think it is natural or human induced?
For that results, we could not say that this. One is the main important thing is climates that drive everything. After that, human activities, and I found that almost the river basins across the Nepal, they have highly human activities from historical period that could make the habitat fragmentations with the human activities. So I blame currently human activity doesn't impact the genetic structures for that.
Okay, there is a question regarding what kind of the food they eat, and it says, can you predict food if the food can alter their genomes, and if there is any alternative food for the red pandas instead of bamboos?
In captive, there are many foods.
Mm-hmm.
In the wild, it's difficult, but I think they eat most of bamboos. In the captive, you can make some habitual for other fruits like that. And I could not give a clear answer for that.
We have another question regarding food again, but from him, Sagar sir. He is asking if food for red panda other than bamboo was paluchinde plant, which you named it on your presentation, was it? Is also a part of the diet, and if so, what importance do you think it is for red pandas in order for consumption?
One of the main things is to maintain bamboo diversity across the range. That is the main thing. Besides that, other food are supplement. Just they contribute very, very low, and that will be used by seasonal. In eastern Nepal, they use such that kind of plant. In central Nepal, I didn't find that plants. In western Nepal, I didn't see that plant. Maybe eastern Nepal, they that bhaluchinde or something, I don't know that name. That could make some supplement food for red panda, not total foods.
Mm-hmm. So in a whole, bamboos are very essential for red pandas diet to conserve red pandas as well. Okay. That was the last of our, we have one more question from Parbat Jang Thapaji, and he's asking, what are the main points to be considered in captive breeding of red panda?
From my study background, we should to manage captive population based on genetic diversity. We need to build more diverse. All these will make this immune system, every system good for that. Maintain genetic diversity in the captive. That will be great for the long run for captive breeding for the population.
Okay.
Besides that, other management issues are many. There are many guidelines for the red panda in the captive.
Uh, Okay, thank you, Dai. And that was the end of our Q&A session. I think we answered all of our questions. Thank you, Arjun Dai, for answering them. Thank you to the audience as well for your questions. It is through your curiosity and questions that really helps us to take the conversation into more interesting way. And so thank you for your participation. Okay, we've almost come to the end of the program now. So before we move on to the closing session, may I ask Arjun to give any final remarks?
I want to say thanks all the participants from Nepal, from abroad, or from all scientists, all seniors, juniors, everyone. As well, I want to thanks for Himalayan Nature, SMCRF, and technical support, NRN, and everyone. I'm very happy, and I give some my findings. I don't know how many people understand it or not, but if difficult, we have a papers, we can go through, they have very details on that. In short period, I could not explain each and everything. So I want to say thanks for all the peoples, all the participants, as well as host and co-host too.
Thank you, Arjun. I'm sure we'll have many people contacting you for future references and more information on Red Panda and your study as well. And thank you for joining us today and sharing your valuable knowledge on Red Panda conservation and what can be done for the future. I'd also like to send my gratitude to the audience for joining in this 18th Nature Speaks series session. I hope it has been an informative evening for everyone. It is through your participation and curiosity in the conservation of nature that has kept our Nature Speaks series going and helping us bring more speakers on board to share their knowledge in this digital platform. So we do encourage you to visit the Himalayan Nature's YouTube channel and Facebook page to check on today's session, as well as previous episodes that has been recorded and uploaded for you to view at your convenience. We will be back with another interesting topic next week, so please do follow the Himalayan Nature's page to find out more about it. And with that, on behalf of the team, I'd like to sign off by thanking everyone again for your presence this evening and wishing everybody a pleasant evening again. Namaste.